Greetings, dear students. Today's lab is about the histology of the respiratory system. Start with the respiratory epithelium. Respiratory epithelium, this is the respiratory epithelium, is a pseudostratified columnar-ciliated epithelium. And as you know, the nuclei appear at different levels. We have cilia on the surface, and of course we have goblet cells. These are the features of this epithelium. And here you can clearly see that goblet cells have typical features. Cytoplasm is clear and the nucleus is at the bottom of the cell. This epithelium rests on a thick basal lamina, as you can see here. So these are the features of this epithelium, which lines most of the conducting zone of the respiratory system. Now we start with the larynx. This is a coronal view of the larynx. We can see cartilages of the larynx. This is the epiglottis. This is the thyroid cartilage. And here we have the cricoid cartilage. You see the staining of these cartilages differ because the thyroid and cricoid cartilages are hyaline, whereas the epiglottis is an elastic cartilage. Therefore, that's why the staining is different. Also, we have these folds of mucosa, pair of superior folds and a pair of inferior folds. Here, you have the ventricular folds and the vocal folds. The space between these two folds is the ventricle. Space between them is called the ventricle. Start with the ventricular fold. The ventricular fold, this fold here, again separated from the vocal fold by the ventricle. If we examine the ventricular fold, the epithelium of this fold is respiratory epithelium. As you can see here, this magnified image, this is the respiratory epithelium with cilia on the surface. The lamina propria is the one typically found under the respiratory epithelium, filled with glands. See here, several glands we can see. And just before we leave this slide, this upward space from the ventricle, this is the saccule, of course. If we look at the vocal fold, this is the vocal fold. Now it differs from the ventricular folds. First of all, the lining epithelium is a stratified squamous non-keratinized epithelium. Remember that these folds are the ones that are responsible for the production of sound, which happens by vibration of these folds. So they require greater protection. This is why they are lined by stratified squamous non-keratinized epithelium. And in the lamina propria of this fold here, there are no glands. If we look at the previous slide, if you remember from the previous slide, we saw how the ventricular folds we had glands. Here in the vocal fold, we do not have glands. But what do we have? Here in this area, we have the vocal ligament. Vocal ligament is formed of dense elastic tissue. And of course, we cannot see the elastic tissue without special staining. So we cannot see it with these normal stains. But this is its position. Also, we have this large muscle, skeletal muscle, the vocalis muscle. This is the vocalis muscle. So these are the features of the vocal fold, lined by stratified squamous non-keratinized epithelium. The lamina propria has the vocal ligament, which is elastic tissue, and also has vocalis muscle, and there are no glands. And as you know, there are also no blood vessels, unlike in the ventricular fold, which has many blood vessels. If you look at this region here, now we can see some glands. Here glands, and this is another gland. Here we can see glands in the lamina propria because at this level the respiratory epithelium has returned. It has returned. So in the ventricular fold we had respiratory epithelium, and then somewhere in the saccule of the ventricle this epithelium has changed into stratified squamous non-keratinized epithelium, and then about this level here the epithelium returned to respiratory. Now regarding the trachea, tracheal wall is formed of mucosa, which is epithelium lamina propria, submucosa, which contains several glands. The third layer is hyaline cartilage, a C-shaped hyaline cartilage, as you can see. And finally, the fourth layer is the connective tissue adventitia. Now remember, posteriorly, the cartilage is absent from the tracheal wall, and instead this gap here is filled with the trachealis muscle, which is a smooth muscle, and elastic fibers. Okay, so the third layer of the wall of the trachea is hyaline cartilage, except posteriorly, where it is occupied by smooth muscle and elastic fibers. This is a magnified view of the wall of the trachea, the mucosa of respiratory epithelium, and the connective tissue under the respiratory epithelium is lamina propria. Then we have some mucosa, another connective tissue layer, usually containing glands. There is a layer of elastic fibers between the lamina propria and the submucosa. Again, this requires a special stain and cannot be seen here in the slide. The third layer is hyaline cartilage. Remember, the hyaline cartilage is surrounded by pericardium on each side. This layer here and this layer here, these are pericardium. Finally, the outermost layer is, of course, adventitia. Now we get inside the lung. First, we see the bronchus. Now, this is a bronchus. Since we can see cartilage, so this is still a bronchus, but we only see a plate of cartilage, This indicates that this is one of the smaller bronchi that are found inside the lungs. If this cartilage was complete ring around the bronchus, this indicates that this is one of the larger bronchi that are found outside the lung. So if I can see a complete ring, this means that this is one of the larger bronchi outside the lung. If I can only see a plate, a small part of cartilage, this means that this is one of the smaller bronchi inside the lungs. Also look at the mucosa. It is folded. This indicates that I have muscles here, because as the bronchial tree becomes smaller, the cartilages decrease, but the smooth muscles increase. And if we go to the next step, the bronchioles, we see first of all, this is section 2 of bronchiole. Again, the mucosa is folded due to the presence of smooth muscles. This is a smooth muscle, and in the bronchioles, the smooth muscles completely surrounds the structure, and there are no cartilages. Once the cartilages disappear, we say that this is a bronchiole. If there is still some cartilage, we say this is a bronchus. Also, the epithelium lining here is no longer respiratory. It is ciliated, simple ciliated epithelium, simple columnar or simple cuboidal ciliated. Also, we can see that goblet cells have disappeared. They start to decrease gradually until they disappeared by the time we reach the terminal bronchiole. And Most of the function of goblet cells and other functions are taken up by Clara cells. So in this magnified view of the epithelium of the bronchioles, this part we can see this is the ciliated epithelium, this is the cilia, these are the cilia. But we also see non-ciliated cells. These cells here, these are Clara cells. These are Clara cells. Now, terminal bronchioles give rise to respiratory bronchioles. Histologically speaking, respiratory bronchioles and terminal bronchioles are alike. So the same epithelium, smooth muscles surrounding the entire structures, of course no cartilages, but the difference between respiratory bronchioles and terminal bronchiole is that into respiratory bronchioles alveoli open. So here we have an alveolus opening into this respiratory bronchiole. This is the main difference. This is the main difference between respiratory and terminal. Into the respiratory bronchiole, and alveoli may open. This is why respiratory bronchioles are part of the respiratory zone of the respiratory system, whereas the terminal bronchioles are part of the conducting zone. So here we can see this is a respiratory bronchiole. Respiratory bronchioles will open into these tubular structures, which are called the alveolar tube. Alveolar tube will end up in an expanded part called the alveolar sac, and into these sacs alveoli open. Of course, alveoli may open directly into the alveolar tubes, and alveolar sacs may open like this part here from the side of the tube. Now all these alveolar tubes, alveolar sacs, and alveoli are simply lined by simple epithelium. Smooth muscles, very few, and sometimes most of the time they are even absent. Mostly we have elastic reticular fibers surrounding the alveoli, but we cannot see them clearly in the slide. Lastly, we see the alveoli. This is an alveolus. Each one of these empty spaces, these all are alveoli. Separating the alveoli is a connective tissue layer, the alveolar septa, which contains several types of cells and fibers. They cannot be clearly seen, of course, under the light microscope. Also, in the alveolar septa, of course, we see the blood vessels, the capillaries. Here are capillaries. These are the capillaries across which gas exchange occur between the alveoli and the blood here at these capillaries. Also in this side here, we see type 1 pneumocytes, simple squamous cells that line the alveoli. Most of the alveolus is lined by this type of cell, simple squamous cells. Also, we have type 2 pneumocytes. These are more difficult to be seen. These are round cells. This could be one of them, but mostly they are not easily seen. The other cell we can see are the dust cell, the alveolar macrophages. The alveolar, this is an example, and this is another example, alveolar macrophages. We see that they have a darker color. The cytoplasm is darker. It is almost brownish. And as you know, these are the cells that are responsible for phagocytosis in the lung, whereas type 1 pneumocytes are the main cells across which gas exchange occur. So this is our lab for today. Thank you and good luck.